Beta-catenin is an evolutionarily conserved protein that performs two conflicting tasks. It functions as the physical anchor for tissue structure and the mobile signal that dictates genetic fate. Historically, research focused on the protein's role as a structural tether within the adherence junction. Later discoveries revealed its secondary function as a nuclear transcriptional coactivator, which moves extracellular Wnt signals into the nucleus to drive the expression of specific target genes. The core functional unit of the protein is a rigid, elongated superhelix composed of 12 armadillo, or ARM, repeats. These repeats fold together to form a central, positively charged groove along the protein's surface. Functionally diverse partners, specifically E-catenin at the cell membrane and TCF factors in the nucleus, utilize this exact same binding site. Because these sites overlap, the two molecules physically collide, creating a state of steric hindrance. This biophysical constraint dictates the protein's regulatory logic. A single beta-catenin molecule cannot multitask. It is physically impossible for it to participate in membrane adhesion and nuclear transcription at the same time. Where the protein is located dictates exactly what it can do. Its influence over cellular life and death is managed through strict spatiotemporal compartmentalization. In a resting cell, the majority of beta-catenin is sequestered at the plasma membrane. It is locked to alpha-catenin near a specific hinge region at residue R151, a conformation required to secure the E-cateherin complex. To mobilize the signaling pool, SRC kinases phosphorylate beta-catenin at the tyrosine 654 residue. This modification reduces its binding affinity for E-cateherin, liberating the protein into the cytoplasm. To determine the spatial requirements for signal transduction, researchers utilized genetically engineered non-degradable forms of beta-catenin and targeted them to specific subcellular compartments. When beta-catenin was anchored to non-adhesive locations like the endoplasmic reticulum or the outer mitochondrial membrane, it still resulted in the accumulation of cytosolic wild-type protein and robust nuclear transcription. In contrast, when the protein was targeted directly and exclusively to the nucleus, it failed to stabilize the cytosolic pool and did not activate transcription. These spatial studies demonstrate that the steady accumulation of the protein within the cytosol is the strict prerequisite for TCF left-dependent transcription in the nucleus. Because free cytosolic beta-catenin drives oncogenic growth, the cell actively suppresses it in the absence of a Wnt stimulus using a multi-protein assembly called the destruction complex. The complex is built on large scaffold proteins, axin and APC, which capture free beta-catenin and position its structurally disordered N-terminal domain for enzymatic modification. Degradation begins with an obligatory priming event. Casein kinase 1 phosphorylates the protein precisely at serine 45. This enables the kinase GSK3-beta to processively phosphorylate the protein at threonine 41, serine 37, and finally serine 33. These modifications create a high-affinity recognition motif for the beta-3CP ubiquitin ligase. The ligase binds to the phosphorylated serine 33 and serine 37 residues, triggering polyubiquination and subsequent destruction by the 26S proteasome. This destruction cycle is interrupted when Wnt ligands bind to frizzled receptors and LRP56 co-receptors at the cell surface. Biochemical analyses support a distributive mechanism of activation known as inactivation by saturation. In this state, the destruction complex captures and phosphorylates beta-catenin, but cannot release it, effectively choking the machinery on its own substrate. Wnt signaling is the targeted, physical jamming of a continuously running molecular shredder, which allows beta-catenin levels to rise and the protein to flood the cell. Beta-catenin evades the destruction complex and enters the nucleoplasm, where it displaces co-repressors. This converts TCF-left factors into bipartite transcriptional activators that drive the expression of cell cycle regulators like c-MYC and cyclin D1. Beyond its role in DNA transcription, recent research shows that beta-catenin acts as a direct RNA-binding protein that governs multiple stages of RNA metabolism. Within the nucleus, beta-catenin interacts with the spliceosome to regulate alternative splicing. In the E1A minigene model, high levels of the protein result in a specific reduction of the 12S and 13S spliced isoforms. In the cytoplasm, the protein binds directly to AU-rich elements in the 3' untranslated regions of specific RNAs. This shields the transcripts from exosomal degradation, increasing the half-lives of cyclin D1 and COX-2 to a degree comparable to dedicated stabilization proteins like QR. The shielding prevents the decay of highly proliferative transcripts. For genes like cyclin D1, the resulting increase in protein production leads to the rapid, exponential cell division characteristic of tumor growth. To counter this, researchers have used cytoplasm-targeted RNA intramers. These engineered aptamers successfully bind to beta-catenin and strip it from the transcripts, restoring their normal degradation. Beta-catenin functions as a scaffold coordinating gene expression at multiple levels, from the initial transcription in the nucleus to the final stabilization of messenger RNA in the cytoplasm. The canonical Wnt beta-catenin pathway is required for mammalian tissue repair across nearly every organ system. In cases of severe toxic liver injury, such as an acetaminophen overdose, beta-catenin stabilization occurs within 1 to 12 hours. This forced nuclear translocation drives the expression of cyclin D1, pushing hepatocytes from the resting G1 phase into the DNA-synthesizing S phase for regeneration. During skeletal fracture repair, the pathway is strictly dose-dependent. Early activation interferes with the lineage commitment of mesenchymal stem cells, while later activation is required for osteoblast differentiation and the mineralization of the new bone matrix. The heart exhibits a biphasic response to injury. Immediately following a myocardial infarction, beta-catenin activation drives the restorative fibrosis necessary to form a dense scar, which physically prevents the heart wall from rupturing. However, sustained signaling in the heart eventually inhibits the differentiation of cardiac progenitor cells and represses the NaV1.5 sodium channel, contributing to adverse remodeling and fatal arrhythmias. Beta-catenin functions as a transient signal. Its precise temporal regulation is the requirement for successful homeostatic repair rather than catastrophic organ failure. Chronic tissue inflammation creates an environment of unyielding Wnt activation. This sustained signal forces resident fibroblasts into hyperactive myofibroblasts. These cells act as biological factories, indiscriminately depositing massive quantities of collagen and fibronectin, which leads to tissue stiffening and fatal organ fibrosis. This can also occur through genetic hijack. Missense mutations at the serine33 or serine45 phosphorylation sites on the CTNNB1 gene abolish the binding sites for the destruction complex. This renders beta-catenin immune to degradation, leading to continuous, unregulated nuclear accumulation. Broad-spectrum inhibition of this pathway is often clinically impossible. Completely blocking the Wnt signal forces the immediate terminal differentiation of LGR5+ stem cells, resulting in the catastrophic collapse of the intestinal crypt architecture. Future pharmacology must focus on precision tools, such as the inhibitor PRI724, which selectively disrupts the interactions between beta-catenin and specific coactivators while leaving homeostatic functions intact. Mapping the exact biophysical switches, the mechanical tension at the membrane, steric hindrance at the arm repeats, and the regulation of alternative splicing is the only way to harness this regenerative potential without unleashing oncogenic ruin.